Hello, dear learner. I, Miss Swapnali Farande from Delonic Society's Baramati College of Pharmacy, Baranpur, Baramati. Today I am going to present new topic Introduction to Bacteria from subject Pharmaceutical Microbiology. Contents of this topic: Introduction to Bacteria, Definition of Bacteria, Structure of Bacteria. After completion of this topic, students should be able to define bacteria, describe the structure of bacteria. What is bacteria? Bacteria are single cell microorganisms that lack a nuclear membrane, are metabolically active and divide by binary fission. These are visible under the light microscope. These belongs to kingdom Prokaryote Monera. They occur in water, soil, air, the ocean, all natural environment and inside the human gut. Definition of bacteria. Bacteria are unicellular organisms belonging to the prokaryotic group where the organisms lack a few organelles and a true nucleus. Another definition of bacteria is bacteria are single-celled organisms without having true nucleus or functionally specific components of metabolism that belongs to the kingdom Prokaryote Monera. This is structure of bacterial cell. Bacterial cell made up of capsule, cell wall, plasma membrane, mesosome, cytoplasm, nucleoid DNA, flagellum, plasmid, inclusion, polyribosome and pilus. In that first one, flagella or flagellum. Flagella are long, slender, thin hair-like structure attached with cytoplasm. They play important role in bacteria for motility. They have 0.01 to 0.02 micrometer in diameter and 3 to 20 micrometer in length. Flagella found in both gram-positive and gram-negative bacteria. Few cocci forms most bacilli and almost all of the spirillum and vibrios are motile by flagella. They can be seen by compound microscope with spatial staining technique and can be easily seen under electron microscope and dark field microscope. Flagella seen in bacterial species in different manner, that is monotrichous, lophotrichous, amphitrichous and peritrichous. Monotrichous means single polar flagella. Example is Vibrio cholera. Then lophotrichous means two or more flagella at only one end. Example is Pseudomonas fluorescens. Then amphitrichous means single flagella or more flagella at both ends. Example is Alcaligenes faecalis. Then peritrichous means several flagella present all over the surface. Example is Salmonella typhi. Then fimbriae. Fimbriae are similar structure like flagella but not involved in motility. It is shorter than flagella, that is 3 micrometer. Fimbriae can be distributed over the entire surface of the cell. Fimbriae act primarily as adhesions and allow to microorganisms to attach to surface. They are responsible for hemagglutination and cell clumping in bacteria. Then piles or pilus. Piles are morphologically and chemically similar to fimbriae. These are present in small in numbers, compatibly fimbriae. Pilus joins to the bacterial cell for transfer of DNA, that is bacterial conjugation from one to another cell. So, pili also called as sex pili or fertility pili, that is F. pili. Then capsules and slime layer. Many bacteria secretes EPS, that is extracellular polysaccharide, that are associated with the exterior of the bacterial cell. The extracellular polysaccharide contain 2% of carbohydrate and 98% of water. So, they produce gummy exterior to the cell. Morphologically, two extreme forms, capsules and slime. Capsules means which forms rigid, tightly and closely associated with cell. and slime, which are loosely associated with cell. Then cell wall. Cell wall gives definite shape to the bacteria. Cell wall is situated between the capsule and cytoplasmic membrane. It is about 20 to 30 nanometer in thickness. The cell wall contains diaminopimelic acid DAP, muramic acid and teichoic acid. These substances join together to form a complex polymer structure known as peptidoglycan or murein or mucopeptide. Peptidoglycan is a large macromolecules containing glycan polysaccharide chain that cross-linked by short peptide bridge. The glycan chain acts as a backbone to peptidoglycan. Those short peptide bridge composed of alternating residues of N-acetyl muramic acid, that is NAM, and N-acetyl glucosamine, that is NAG. Each molecule of NAM attaches a tetrapeptide. Tetrapeptide consisting of the amino acids L-alanine, D-alanine, D-glutamic acid, lysine or diaminopimelic acid, that is DAP. Cytoplasmic membrane. Cytoplasmic membrane is thin near about 5 to 10 nanometer. Biochemically, the cytoplasmic membrane is fragile phospholipidic bilayer with proteins distributed randomly throughout. In the phospholipids bilayer, most of the proteins are tenaciously held and are called integral proteins. Other proteins are loosely attached, those are called peripheral proteins. The phospholipid molecules are arranged in two parallel rows called a phospholipid bilayer. Each phospholipid molecule contains a polar head and a non-polar tail. Polar head composed of a phosphate group and glycerol, and the non-polar tails are interior of the bilayer. Prokaryotic plasma membrane are less rigid than eukaryotic due to lack of sterols. Functions of cytoplasmic membrane are the including in transportation of nutrients, it provides mechanical strength to the bacterial cell, it helps in DNA replication, it contains the enzymes involved in the biosynthesis of membrane lipids and various macromolecules of the bacterial cell. Then the cytoplasm. The bacterial cytoplasm is a type of suspension in that contains organic, inorganic solute in a viscous water. It contains the nucleoid, ribosomes, proteins and other water-soluble components and reserve material. The cytoplasm bacteria differ from that of higher eukaryotic microorganisms is not containing endoplasmic reticulum, Golgi apparatus, mitochondria and lysosomes. In most of the bacteria also contains extrachromosomal DNA, that is DNA are not connected to chromosomes, is also present. Then ribosomes. Ribosomes are most important structure in bacterial cytoplasm. They involved in protein synthesis. Ribosome numbers varies with the rate of protein synthesis. If greater the number of ribosomes, then the greater the protein synthesis. They have 25 nanometer in diameter. They are characterized by their sedimentation properties. These bacterial ribosomes are called as 70S ribosomes. Then mesosomes. In most of the bacteria, particularly in gram-positive bacteria, the growth condition depending upon the membrane appear to be infolded at more than one point. Such infoldings are called mesosomes. Mesosomes present in two types, central septal mesosomes and peripheral lateral mesosomes. Central mesosomes present deep into the cytoplasm and locate near the middle of the cell. These are involved in the DNA segregation and in the formation of cross-wall during cell division. And the peripheral mesosomes are not present at the central location and are not associated with nuclear material. Mesosomes are also called as chondroids and are visible only under electron microscope. Larger number of mesosomes have a higher respiratory activity. Example is Azotobacter. Then spores. Many bacterial species produce spores inside the cell and outside the cell. Inside the spores are called endospores and outside the spores are called exospores. Examples are Bacillus anthrax and Bacillus subtilis, etc. Spores are extremely resistant to desiccation, staining, radiation, disinfecting chemicals, and heat. Each bacterial spore on germination forms a single vegetative cell. They remain visible for a long time and help bacteria to survive for a long period under unfavorable conditions. The spore envelope consists of the inner membrane, outer membrane, cortex, and spore coat. In some species have the outer layer called exosporium which bear ridges and fold. These are some references for this topic. Thank you.